Hi everyone, I'm Xiang, the first author of this manuscript. It's my pleasure to share our recent work on Golgi remodeling during paraptosis. Paraptosis is a type of non-apoptotic programmed cell death. As shown here, its main features include cytoplasmic vacuolation, ER dilation, and mitochondrial swelling. But most studies have focused on the ER and mitochondria, while the Golgi's role has been less clear. In this study, we focused Golgi remodeling during paraptosis and identified a structure that hasn't been described before. Then, during our analysis of organelle changes, we noticed something interesting. Besides the typical low-density vacuoles induced by glavridin, we also observed dense and round structures inside the cells. Importantly, these structures were not unique to glavridin. We saw the same structures after treatment with two other paraptosis inducers, morlucin and honokyo. We call these structures paraptosomes as a reproducible feature of paraptosis. To understand the origin of these structures, we analyzed different organelles. The most striking change was observed in the Golgi apparatus. Normally, the Golgi forms a compact structure near the nucleus, but during paraptosis, it fragments and reorganizes into these circular patterns. It closely matched the dense paraptosomes we observed. We next used electron microscopy to examine the ultrastructure of paraptosomes. These structures contained densely packed, membrane-rich materials arranged in a complex network. Combined with organelle marker analysis, these results suggest that paraptosomes are distinct from conventional mitochondria, lysosomes, or autophagosomes. During paraptosis, Golgi stress markers were activated, while VEGF-A secretion was reduced based on ELISA analysis. Normally, the cleaved oasis N-terminal fragment enters the nucleus. However, during paraptosis, it accumulated around the paraptosomes instead, indicating impaired Golgi nuclear signaling. These findings suggest that Golgi disintegration impairs protein trafficking and stress signaling. In summary, we found the paraptosomes are Golgi-related structures that consistently appear during paraptosis. The Golgi changes early on, and this messes up cells' normal trafficking and secretion. So these findings indicate the Golgi disintegration is a brand new feature of paraptosis, but we still need to figure out exactly how paraptosomes form and whether they actually play a part in causing cell death. Thank you.